Here we have that family tree of all life on Earth. We're visiting this again, so you can see that here, at this point, that's when life started, and that's where you would find Luca, the last universal common ancestor. Directly stemming from that ancestor is this group here, bacteria. And we'll first talk about bacteria and archaea, and then move on to eukaryotes. So if we were to compare the timeline of Earth and life on Earth with a road trip, it would go something like this. We would start our trip up here in Canada at Kamloops. Now this is the beginning of Earth about 4.6 million years ago. We'd head south from there, and we'd have no life on Earth for several hundred miles. Our oldest dated rocks formed about 3.8 million years ago. As we continued south, we would finally hit our evidence of the first life forms on Earth, and that would be somewhere between 3.5 and 3.8 million years ago. Those took the form of prokaryotes, and we'll keep going south from there. As these prokaryotes metabolize and reproduce, they're introducing more and more oxygen into the air. And so this would eventually lead to one of the first mass extinction events, where oxygen, which is toxic to a lot of those early organisms, would kill off a lot of the early diversity. We wouldn't actually find eukaryotes until about 2,800 miles into the journey. That would be about Oklahoma City. Finally, we wouldn't even find multicellular organisms until about 3,400 miles into the journey, about 2/3 of the journey. Finally, we'd see this explosion of diversity about 4,000 miles in, and we would see plants and other animals colonize land about 4,200 miles in, or 500 million years ago. We would see dinosaurs at the tail end of the journey, about 180 million years ago, and we wouldn't see Homo sapiens until the very end of the journey. The last few blocks we would find Homo sapiens, about 4,600 miles into the journey. This is important because it shows you that life occurred very early on in Earth's history, and a majority of that time was spent in a unicellular world. A lot of those organisms that dominated the timeline of Earth were unicellular in nature. And they included prokaryotes and some eukaryotes that you might be familiar with. Finally, it wasn't until very late in this timeline that you had organisms colonize land and the development of what represents a lot of what you might think of as animal diversity. These early lineages are dominated by two groups, the bacteria and archaea. Bacteria and archaea are prokaryotic. They have a circular chromosome. They're mostly unicellular, and some can fix nitrogen. Many species of archaea can also grow at really extreme temperatures. One notable and kind of useful feature of bacteria is the presence of plasmids as genetic information inside their cell bodies. Now, outside their bacterial DNA, you'll often find these rings of genes. These rings often contain genetic information for the production of byproducts that can be useful to the bacteria under certain conditions. So this is kind of crazy. That means that when bacteria need a certain piece of genetic information, they can sometimes find that outside of their genome, incorporate that when they need it, and start creating those byproducts as needed. That's kind of like having a toolkit or a tool belt filled with different genes, and when situations get stressful or require some new ability, you can acquire that new ability by pulling out those genes and incorporating them into your genome. This is something that we've taken and used to our advantage to create byproducts when we need them from the bacteria. In this example, we're looking at a plasmid. And remember that plasmid is a ring of genetic information that's somewhere inside the cell body of a prokaryote, of a bacteria. So here's the plasmid, and what we're going to do is we're going to insert an insulin gene, a gene that makes the product insulin inside this plasmid. Now, using several different genetic processes, what we end up with is what we call a recombinant plasmid. That's a plasmid that we've recombined with a gene that we find useful. In this case, that's insulin, which would be used in the medical field. So once we create those recombinant plasmids, we can then introduce them into an environment or solution with those bacteria. Then you want to do something to shock the bacteria and damage their cell wall in order to allow those plasmids to enter the bodies of the bacteria. In this case, we're doing a process known as heat shocking. So we heat shock those bacteria. We'll actually create little holes, little damaged points on their cell bodies, and the recombinant plasmids will then enter the bacteria. So now we've introduced genetic information as plasmids into the cell bodies of some of those bacteria. Not all of the bacteria will take those up, though. So once we've done the heat shock treatment and those bacteria have taken up those plasmids, we need a way to get rid of the bacteria that don't have the plasmids and only keep the ones that can make the gene product that we want, in this case, insulin. So when we made that recombinant plasmid, we also introduced another gene, not just the one that produces insulin. We also introduced a gene that is coding for antibiotic resistance. The bacteria that took on those plasmids that can now produce insulin also, in taking up those plasmids, took up the ability to resist antibiotic. So now those bacteria should be resistant to antibiotics. Next, you grow the bacteria, all of them, on a plate of antibacterial. Only the bacteria that took on those plasmids that have the antibiotic resistant gene will continue to grow. And so now you know that those bacteria also produce insulin. You can select from those colonies and reproduce those colonies to produce insulin. While that insulin byproduct isn't a finished product, it is a great head start and allows us to grow insulin using bacteria